Okay, good afternoon.
Good afternoon po, Doc.
Can you hear me? Can you see my screen as well?
Yes po, Doc.
Can you hear me?
Yes po, Doc.
Can you see the screen? I think I have allowed to unmute. Di pa rin. Can you try unmuting? Could you try unmuting? I think it's okay.
Doc, okay na po.
Okay na po daw.
Can someone try unmuting?
Okay na po daw.
Okay na po, Dok.
Hello? Can you unmute?
Po daw.
Doc, okay na po.
Ayun,
Nakapag-unmute na.
ako pala ang may problema. I'm sorry about that. Okay. Can you see the screen?
Yes po, Doc.
Okay. So, for this afternoon, what we will have would be gene regulation. Okay. So, what is your understanding with regards to regulation? What is being regulated? What do you know of? Not necessarily the genes. What are the regulations that you would know? And what is it for? Anyone? What is regulation? What's being regulated? For example, you as a medical student, how do we regulate you? Anyone? Kailangan ko pa ba magtawag? Wala pa. Simple pa tanong ko. Zelan Tobias. How are things regulated? Ano bang mga hilig nyo? pa ako. Anyone?
Up to control po. Regulate to control.
For control of whatever. For example, as a medical student, okay, what is your means for control? Or what would be our means for controlling quality, quantity? So for example, pagpasok nyo pa lang, what are the requirements for you to enter medical school of perpetual?
NMAT po, Doc.
NMAT, okay? NMAT of what?
50 po and above.
NMAT of 50. Although some would have waivers of NMAT less than 50, but would have to have no failing subjects. So that's a form of regulation. What else? As you go through your medical career, what are the other regulations that you will encounter? For first year, what are your regulations? Exams. And then come second year, for you to go third year, there's seminar 1, and then seminar 2, then clerkship. Then of course, the penultimate boards. But it doesn't stop there. You have your residency, diplomate, etc., etc. And that would set different steps of regulation for you to become the doctors that you want to be. And why do we need to regulate? Why is there a need to regulate, for example, you as medical students?
Doc, to be assured po of the quality.
So to ensure quality, that's correct. And to know, to regulate quantity, in the case of genetics, you will encounter feedback mechanism. So what would your feedback mechanism tell you? How does it work?
The areas that you need improvement pa po.
So your feedback in such a way that if, for example, madami na siya, you will send signals to your body that you have enough of this enzyme or substrate, etc., you will stop producing. Same way, if it's konti or if it's scarce, you will send signals to your body as well to send signals that you have to produce more to compensate for scarcity. And that is how our body works. So for example, feedback mechanism in medical school, if you know that you are good standing in a certain subject or kayang-kayang ipasa, so you sacrifice some of that so that you put more effort into producing points, for example, from anatomy or biochemistry. Okay. Something like that. and that is regular. So goals for this lecture would be to learn DNA elements that control transcription, your protein factors that control transcription, your lac operon, which would be the model for your regulatory gene, and regulation of protein synthesis by RNA. Okay. So these are the different ways that we could regulate protein. So at the entry level, so sa start pa lang, you're being regulated. And then midway, post-transcriptional, transport, degradation, every step, there is a certain form of regulation in biochemistry. All right? Before, it was initially focused on transcription initiation only. However, with the advent of more data or of more studies, post-transcriptional and translational regulation are more understood. Hence, it is more studied and different mechanisms are elaborated, especially in the development. Okay? Such that you would see regulation in different DNA and different protein amino acids, which we will see later. Okay. So the basics of it. So it's a housekeeping gene. So it's constantly expressed as in all cells. So there is an inducible and a repressible. So if it's an inducible gene, it turns on the operon. We will see what the definition of operon is later. And repressible, you are able to turn it off. Okay. So there is such a thing called promoter. So it is found near the starting point of any transcription initiation. And this interaction greatly influences the rate of initiation of transcription. Okay? So this enhances or inhibits this interaction between your RNA pol and the promoter DNA. So far you have your gene, which is repressible or inducible, and your promoter, which is the signal of the which is the starting point of your gene transcription. Okay. So here you will see, so this is just in E. coli. So this is the sequence wherein most bacterial promoters usually are in the 10 and the 35 regions, interact with the sigma factor of your RNA polymerase. So substitutions in this 10 and 35 region would usually reduce the affinity of your RNA pol for the promoter. Okay, so we have the sequence, your promoter and your inducible and repressible gene. So some promoter would also have the upstream element that interacts with your alpha subunit of your RNA polymerase. So this is your, oops. So this is your 5' N. So this is your 10 and 35 region. So you have your upstream element as your sequence. Okay. What else would be included? So there are mechanisms to regulate transcription in bacteria. So these sigma factors function as a way to recognize different classes of promoters. And this allows coordinated expression for different sets of genes. So other functions would be binding other proteins to the promoters, such as the following. Okay? So there's regulation by specificity factors for the sigma subunits. For example, your E. coli, if subjected to heat, your sigma 70 will be replaced with sigma 32. And what will happen, this will direct your RNA pol to different promoters and hence have different results. But still protect the E. coli as a structure that even in heat, it doesn't lose conformation. So this heat shock induces transcription. So it changes from your sigma 70 to 32 and it will change your promoter and that would change the transcription sequence. Okay? So this would be repressor and effector. The difference of which repressors would reduce your RNA pol promoter interactions or block the polymerase. So this is usually near a promoter in bacteria but further away in many eukaryotes, which we will learn more about in the latter slides. Your effectors can bind to a repressor and induce a conformational change. So remember, what could induce a conformational change? Covalent or non-covalent bonding. So it could either be one or the other, but it induces a change so that your repressor could be more effective. So this could be either increase or decrease your affinity for the operator and thus increase or decrease transcription in the process. So these binding sites in DNA for activators are called enhancers. So enhancers are usually adjacent to the promoter. So often adjacent to promoters that are weak, so the activator is necessary. Think of it, so if you are just, for example, in clerkship and internship, you are just a clerk. You are often paired with either another clerk or another intern if you cannot work alone. So sometimes that is needed. So in eukaryotes, enhancers may vary or may be distant from the promoter. So what do we have so far? Ano ang magkakalapit? Promoter, what else? Enhancer, you have your repressible gene, either inducer or repressor, your activator, your effector, and repressor. Okay? Yan ang mga magkakasama. Okay. So, we go back to the concepts of negative regulation and positive regulation. So, example of which repressor binds to DNA and shuts down transcription. So, you will look at these two examples. Despite the opposite effects, the operon turns on or the operon turns off, both are considered negative regulation. So, the first example, your molecular signal causes dissociation of your repressor from your DNA. So, natanggal si repressor. So, because your repressor gene is removed, your operon is on, thus inducing transcription. Okay? Another negative regulation, your molecular signal causes binding of your repressor to your DNA. It binds to your DNA. So thereby, so this repressor turns your operon off, but is also considered negative regulation. Same way, your positive regulation. So it usually involves activators. So what are activators? They enhance the activity of your RNA polymerase. So your activator binding sites are usually near your promoters, weakly binding to your RNA pol, or do not bind at all. So even if these two would have different end results, this would be examples of positive regulation. The first one, your molecular signal causes dissociation of activator from DNA, thereby inhibiting transcription. Your second, it causes binding of your activator to your DNA, thereby inducing transcription. Okay? So there is a concept such as DNA looping. And this is how, even far away, you could influence transcription. The promoters can influence transcription thousands of proteins away. So remember, if you are making a coil or a spiral, the distance, even if it's kilometers away, if we put it in a loop, it will be shorter. So this would be facilitated by your regulator proteins with your coactivators. And what will happen? The distance would be shortened. Hence, even if your protein, your promoter is around 30, 40 amino acids away, it could still enhance these, affect these proteins. Okay. So this is the structure of your operon. So remember, you have your promoter, your operator, the genes that needs to be transcribed, and activator, and a repressor binding site. So this is the most common used model of your gene expression. This is your lac operon. And it's a cluster of genes sharing a promoter and regulatory sequences. Okay? And these are your components. So the lac operon was discovered 1960 through the work of Jacob and Monod. So knowing the three genes for metabolism of lactose that works together in an operon. So know your LAC-Z, LAC-Y, and LAC-A. So LAC-Z is your beta-galactosidase, and LAC-Y is your lactose permease, and LAC-A is your thiogalactoside transacetylase. So your LAC-Z, it cleaves lactose to yield your glucose and galactose. So your lactose permease or your LAC-Y is your transportation means of lactose into a cell. So they rely heavily on negative regulation via a repressor. Okay, so know these concepts. So when glucose is abundant and lactose is lacking, you know that glucose is the primary source of energy for our body. Of course, your lactose metabolism would be repressed. If glucose is scarce and lactose is present, then the cells can use this as an energy source. So the cells would suddenly have the need to express the genes for enzymes for lactose metabolism, such that the repression is removed. Okay, thereby allowing our body to have lactose metabolism to be dominant. Okay, this is your lactose metabolism in E. coli. So again, your beta-galactosidase, your LAC-Y, lactose permease, and your LAC-A. Okay. what else do you need to know? So you have a LAC-I, that is your initiator, encodes a repressor called the LAC repressor. It has its own promoter, P initiator. So transcription of your repressor is independent of transcription of the enzymes the repressor regulates. So then it binds to O1, O2, or O3. So the LAC repressor primarily binds to your O1 because O1 is adjacent to the promoter. So binding of the repressor helps prevent your RNA polymerase from binding to the promoter. What then happens? Once it's bound to O1, it will then either bind to O2 or O3 depending on the looping and where it will end up. Okay? So this is your LAC-I and your P initiator. This is your O1, your LAC-Z, LAC-Y, and LAC-A, 3' and 5' end. So this is your O1. So it binds to your O1. What will then happen? Once it binds with your O1, your promoter will then form a loop, either binding it to O2 or O3. In this case, it binds with your O3 because it made the promoter near the O3. Hence, forming a loop between the two O1 and O3. Okay. So, lac operon is also induced by alleolactose. So, it binds to the repressor and causes it to dissociate from the operator. So your beta-galactosidase not only hydrolyzes lactose, but it can also isomerize lactose into alleolactose, such that the relationship would be directly proportional. So your lac repressor binds to DNA, structure it's a tetramer, and as mentioned, your O1 sequence reflects the symmetry of the repressor. But it's not as simple as that. Remember, our body has other alternative source of energy. So it's not just the presence or absence of glucose that would govern the expression of lactose metabolism. So remember, you have your cyclic AMP and your cyclic AMP receptor protein, your CRP or CAP, or that's your catabolite activator protein. So what is this? So your CAMP binds near the promoter, o dagdag na sila doon. So it simulates transcription 50 times. It bends your DNA and it doesn't form readily without your CRP and CAP. So it has only this effect when the lac repressor has already dissociated. So CAMP appears when glucose is low. Okay? So when glucose is high or low but lactose is absent, repressor stays bound. So no transcription even when there is CRP or CAMP. So glucose high, CAMP low, or glucose low, CAMP high, but lactose is absent, no gene expression will take place as far as the lac operon is concerned. Okay? But when your repressor dissociates, but transcription is only stimulated significantly if your cyclic AMP rises. So glucose is high, CAMP is low, lactose is present, your repressor is dissociated, there is a low level of gene expression. However, remember your CAMP again multiplies your transcription 50-fold. Thereby, if CAMP is high, lactose present, glucose is low, there is high level of gene expression in your lac operon. Clear? Okay. So in summary, the strongest requirement for induction of the lac operon, your lactose must be present, thereby reducing repression, and your glucose must be so low that CAMP needs to intervene, bind to CRP, form that complex, and be an activator. So two requirements for your strongest induction would be presence of your lactose and a very low glucose. Okay? And more or less, this summarizes the combined effects of your glucose and lactose on the lac operon. Low, it's inhibited. Lactose is high, permits transcription. Glucose is high, transcription is dampened. And glucose is low, CAMP forms, thereby it's activated. The last two slides, be clear on that. I want you to familiarize yourself with these relationships. Okay. So it also involves hydrogen bonding. So glutamine and asparagine can form a specific hydrogen bond with your adenines. Arginine can form specific hydrogen bonds with your cytosine guanine. So this could be seen on your later slide. So this is the visual representation of the previous slide. So this is an example of your DNA protein binding. So other concepts that I want you to take note of would be your protein arrangements used in thousands of different proteins. I do not expect you to know the loops, know the combination, etc., etc. Suffice it to say, I want you to know that these are the protein DNA binding motifs. So, important to take note of would be the helix turn helix. This would be used by your lac repressor gene, your zinc finger, and your leucine zipper. Okay. So your helix turn helix is approximately 20 amino acids. So there's one alpha helix, then one beta turn, then another alpha helix, which you would see in the later slide. So this is a sequence-specific binding due to specific contacts between recognition helix and the major groove. Okay. So your alpha helix is red, then your beta turn is blue, and your alpha helix again. Okay? So your zinc finger, so the finger portion is a peptide loop cross-linked by your zinc ion. So usually this is 4-cysteine or 2-cysteine, 2-histidine. So this interacts with your DNA or RNA. Binding is weak. So several zinc fingers often act in tandem. So these are usually your nuclear repressors. So this is an example of your zinc finger motif. You will see 3 zinc fingers of the regulatory protein. So each zinc coordinates with 2 histidine and 2 cysteine residues. So the leucine zipper motif, so it's a dimer of 2 amphipathic alpha helices plus a DNA binding domain. So One side is hydrophobic, one side is hydrophilic. So approximately every seventh residue is leucine. That's why it's called Elastin Zipper. So these helices form a coiled coil. So this one would have residues from lysine to arginine to interact with your polyanionic DNA. Okay? This is your structure of a leucine zipper from yeast. So the zippered alpha helices would be gray. So this would be derived from different subunits of your dimeric protein. Then the two helices wrap around each other, so forming a coil. Thus, the interacting would be the leucine side, and the conserved residues are colored to correspond to the sequence as such. Okay. So your eukaryotic gene regulation relies on combinatorial control. So in yeast, there are only 300 factors for thousands of genes. So they mix and match different combinations to create different genes and would depend on these different protein-protein interactions. Okay. So another concept is your RNA binding domain. So these are your RRNs. So it's getting longer. So now they are 90 to 100 amino acids. So this would be the 4-strand antiparallel beta sheets with 2 alpha helices. So they're usually found in gene activators and bind to both DNA and RNA. Okay. So these are examples of your RRM. Okay. So amino acid biosynthesis is regulated by transcriptional attenuation. So here we will see our other Operon model, which is tryptophan. So this is regulated by transcription attenuation. So transcription begins but is halted by a stop signal, that is your attenuator. So it relies on the fact that in bacteria, transcription and translation can proceed simultaneously. So usually it's in the 5' region of a leader sequence and it can make the ribosome stall. So the attenuator, which is part of the leader, determines if the transcription will be initiated or halted. Okay. Your attenuator is your purple, your leader is your light blue. Okay. So what happens? This operon would be regulated by two mechanisms. So when tryptophan levels are high, the repressor binds to its operator. Here is your operator. And transcription of your tryptophan mRNA is attenuated. So the biosynthesis of your tryptophan by the enzymes encoded in the TRP operon is diagrammed at the bottom. Okay? How does it know if it will attenuate or proceed with the interaction? So the leader is 162 nucleotides long and has segments 1 to 4. So 3 and 4 would form a base pair and they form a hairpin structure. That is the attenuation signal. So 3 and 4 would be your stop signal. If 2 and 3 base pair forms, that is your transcriptional signal, thereby no attenuation. So how will the opera know if it would be 3 and 4 or 2 and 3? So these are the 4 segments, 162 amino acids. So transcription is initiated at the beginning of your 162 amino acids. So it's regulated. So it would either be 3 and 4 or 2 and 3. So that is your 3-4 pair and that is your 2-3 pair. How will it know? So segment 1 is immediately translated. So regardless, your segment 1 translated, and because the ribosome is closely behind your RNA pol. So segment 1 contains the important TRP codons. Okay. So here is where it's amazing. If your tryptophan is abundant, translation proceeds so that segment 2 is covered with ribosome and can't pair with segment 3. So if it cannot pair with segment 3, it will then pair with your segment 4. Then it is attenuated. No more further production of your tryptophan because tryptophan is in abundance. On the other hand, if it's scarce, the ribosome wall will stall at TRP codons in your mRNA. This allows pairing 2, 2, and 3. Translation then proceeds and proceed to producing more tryptophan. This is just a model. Your other amino acids also operate this way. So leucine also works this way, histidine, phenylalanine, etc. So don't get confused if it gets substituted. Ay, hindi TRP, hindi tryptophan. Baka hindi pwede yung ganito. So, some examples wherein this tryptophan model could also be applied. Okay? So, this is what happens when TRP synthesis is not needed. When it's not needed, it's attenuated, your base 2 and 4 will form. When synthesis is needed, your base 2 and 3 will form and therefore produce more tryptophan. Okay? Furthermore, a prepressor protein also regulates your TRP transcription. So, this repressor is a homodimer. When TRP is abundant, it binds to your repressor, causes it to bind to the operator, and slows expression of genes for TRP synthesis. So it is not just your 2,4 which attenuates your TRP production. There is also a repressor gene that binds to your operator to stop expression of TRP synthesis. So this homodimer has helix turn helix motifs that interact with your DNA via a groove. Okay. So another concept would be your SOS response. This would usually be in response to DNA damage. So normally these genes are repressed by the LEX-A repressor, and your LEX-A binds to operators at several genes. So what happens would be damaged DNA would produce a lot of single strands. So these are your ssDNA, which is eventually bound to your REC-A, or in eukaryotes, your RAD51. Okay? So your REC-A binds to your LEX-A repressor, causing it to cleave and dissociate from DNA. So REC-A is also considered a co-protease. Okay. So remember, your damaged DNA would produce your single strand, ssDNA. Then this would bind to your single-stranded DNA, that is your LEX-A, thus your LEX-A repressor is inactivated. Okay. So some repressors keep viruses in a dormant state within the bacterial host. So your REC-A or RAD51 can help cleave and inactivate these other repressors. And sometimes when it's in ORI, when it's damaged, it allows your virus to replicate, lyse cell, and release new virus particles. So when bacteria would need more protein, so they make more ribosomes, and this consumes energy and resources, thereby it's also a highly regulated process. So these ribosomal protein operons are regulated via translational feedback. So what is translational feedback? So each operon for an R protein encodes a translational repressor. So a repressor binds to your messenger RNA and blocks translation. So your repressor has greater affinity for your rRNA than for your mRNA. So translation is repressed only when synthesis of R proteins exceeds a level needed to make ribosomes. So for example, the R proteins that act as translational repressors are shaded light red. So each translational repressor blocks the translation of all genes in that operon by binding to the indicated site of your mRNA. So genes that encode subunits of RNA polymerase are shown in purple. Okay? So genes that encode elongation factors are blue. So the R proteins of the large ribosomal subunit are designated L1 to L34, those of the small S1 to S21. So there is called astringent response. This occurs when your amino acid concentrations are low. So when there is lack of amino acid, thereby it produces uncharged tRNA. So rRNA synthesis triggers a cascade that begins with binding structure, binding stringent factor protein to ribosome. Okay? So your stringent factor catalyzes formation of your nucleotide, guanosine tetraphosphate. So it is formed from adding diphosphate to the 3' end of your GTP. Then a phosphorylase cleaves a phosphate to yield your guanosine tetraphosphate. So your binding of your guanosine tetraphosphate to your RNA polymerase reduces your RNA synthesis. Okay. This is your stringent response in your E. coli. So this is a response to your amino acid starvation, which is triggered by binding of an uncharged tRNA in the ribosome site. So the stringent factor binds to your ribosome and catalyzes the synthesis of your guanosine tetraphosphate, which is converted. So this signal reduces transcription of some genes and increases that of the others. Okay. So remember your cis and trans. Cis would be its own function. Trans would be a separate molecule. So example of which would be your mRNA of your gene R polymerase encodes a sigma S, a specificity factor used by E. coli in stress conditions. So such as starvation, when your sigma S is needed to transcribe stress response genes. So this your sigma S is present at low levels but is not translated due to a hairpin structure that inhibits its binding to ribosomes. So your small RNAs bind to this mRNA and inhibit formation of the hairpin. So the small RNAs would include your dsRA and your rpRA, which would be then facilitated by a chaperone protein called HFQ. Okay. So this is how your regulation of bacterial mRNA function in trans by sRNA. So several sRNA would be involved in regulation. So they would all require the protein HFQ. So this is an RNA chaperone that facilitates RNA and RNA pairing. So what happens is your dsRA promotes translation by pairing with one strand of a stem loop structure that would otherwise block the ribosome binding site and others. So RPRA is similar but is not shown in this diagram. Okay. So this ribosome binding Schindel-Garno sequence is sequestered into a stem loop structure in the mRNA. So in the presence of your HFQ, your small regulatory RNA dsRA binds to your mRNA. So this binding opens the stem loop and allows mRNA binding to the ribosome. Thereby, the sRNA promotes translation. On the other hand, in the ribosome binding Schindel-Garno sequence in a stem structure, small regulatory RNA OxyS binds to your mRNA. So the binding of OxyS blocks the ribosome binding site in your mRNA. Thereby, OxyS RNA inhibits translation. Okay. So there would be such a thing called riboswitch. So this is domain of an mRNA that can bind a small molecule ligand. So this binding of ligand would affect conformation of the mRNA and its activity. So it's illustrated by several different riboswitches based on a widespread natural aptamer that would bind thiamine pyrophosphate, and TPP binding to the aptamer would lead to a conformational change that produces the varied results illustrated in parts. So B and C would be different systems in which the aptamer is utilized. So it's a developing area of research and it's found to respond to many coenzymes found in your eukaryotic introns, and some are unique to bacteria and are targets for antibiotics. Okay. You have heard of gene recombination. So these are processes that can remove promoters relative to the coding sequence or can put genes into multiple orientations to alter the expression. So in one orientation, FLJB is expressed with a repressor for FLJC gene. In another orientation, only FLJC is expressed. This is called phase variation. Okay. So the products of your genes, FLJC and B, are different flagellants. The HIN gene encodes the recombinase that catalyzes inversion of your DNA segment containing the FLJB promoter and the HIN gene. So the recombination genes are called HICs. the ones in yellow. So in one orientation, B is expressed along with a repressor protein. So that represses transcription of the C gene. So in the opposite orientation, only the C gene is expressed. So the A and B genes cannot be transcribed. This interconversion between these two states is known as phase variation and also requires two other non-specific DNA binding proteins. So these are examples of gene regulation by recombination. So other features of your eukaryotic gene regulation would be the access of your eukaryotic promoters to your RNA polymerase, sometimes hindered by chromatin structure. Thus, it requires remodeling of your chromatin. So positive regulation mechanisms predominate and are required for even a basal level of gene expression. So it usually requires a complicated set of proteins. So there are usually three features, your euchromatin. So this is your less condensed chromatin. And chromatin remodeling of transcriptionally active genes. So it's either your nucleosomes are repositioned, there are histone variants, and there are covalent modifications to nucleosomes. So you have your SWI switch or your SNF or your sucrose non-fermentable complex. So this remodels chromatin to irregularly spaced nucleosomes, stimulates binding of your transcription factors, works with proteins of your imitation switch family, it's ATP dependent, and so on. Okay. So there's remolding. Eukaryotic cells generally have 9 to 10 different chromodomain helices grouped into 3 subfamilies. So each family would have a specialized role. For example, INO80 family remodels chromatin and DNA repair, and etc. So, covalent modification of histones would be done by the following. So, it could either be methylated, phosphorylated, acetylated, ubiquitinated, sumylation, and this occur mostly in your N-terminal domain of the histones found near the exterior of the nucleosome particle. Okay. So, covalent modification allows recruitment of enzymes and transcription factors. So, your methylation of your lysine 4 and lysine 36 at histone 3 and arginine of histone 3 and histone 4 would result in transcriptional activation. So, this recruits histone acetyltransferases and then activate a particular lysine. So reversed by histone deacetylase that makes chromatin inactive. Thereby, acetylation of your lysine results in decreased affinity of histone for DNA. So this would be some enzyme complexes that catalyze chromatin structural changes associated with transcription. Okay? So there's a nucleosome code or histone code wherein genomes encode directions for nucleosome organization. So nucleosomes seem to occur at specific sequences, and there are covalent modifications seem to occur at specific regions. So a nucleosome positioning code or histone modification code could explain these observations. Okay. So positive regulation could occur when they are dependent on activator proteins and not RNA pol dependent. Most promoters are inaccessible, thus making repressors redundant. Combinatorial control provides a more precise positive control for gene regulation, and negative regulation exists but typically involves LNCRNAs and not proteins. So RNA polymerase 2 requires 5 types of proteins. So these are your transcription activators, architectural regulators that facilitate looping, chromatin modification, co-activators, and basal transcription factors. So enhancer proteins first are diverse. So they can bind thousands of nucleotides away from the data box of the promoter. So they can have DNA binding, protein binding, or signal molecule binding domains. So some could regulate a few genes, some may regulate a number of genes. The second would be your architectural regulators that regulate looping. Again, DNA looping allows distant enhancers to affect far away proteins. So example would be the high mobility group of proteins which have multiple functions including architecture regulation. So coactivators also assist in polymerase. So your mediator complex binds to your carboxyl terminal domain of your RNA pol2. So these are required for your both basal and regulated transcription at many promoters. So your first component of pre-initiation complex is your Tata box or your Tata binding protein at the typical Tata box of a promoter. So other details of eukaryotic regulation that are emerging, so your binding of activators triggers many promoters to bind RNA polymerase 2. So binding of one activator seems to enable binding of additional activators. Histones are displaced as activators are bound, and the process coordinates with chromatin remodeling. This could be seen in yeast galactose metabolism, so the regulation of genes for importing and metabolizing galactose in yeast illustrates important principles. So these are spread over several chromosomes but have similar promoters. So these GAL genes are regulated by a common set of proteins. So GAL3P forms a complex with your GAL4P and GAL80P and allows your GAL4P to be the activator. GAL4 also recruits the sweet and the non-sucrose factor and mediator complexes for opening the chromatin. So these are the examples of genes of galactose metabolism in yeast. So other features of your hormone-mediated regulation would be your hormone receptor complex binding to your DNA regions called your hormone response elements. So your hormone receptors have a DNA binding domain with your zinc fingers. So your hormone receptors also have a ligand binding region at the C-terminus that is highly variable between different receptors. So there are two types. So your monomeric type 1, so these are usually receptors for your sex hormones and glucocorticoids. This would be found in your cytoplasm, and when the hormone binds, your HSP70 dissociates. So what happens is your receptor dimerizes and exposes your nuclear localization region such that your complex travels to the nucleus to be the transcriptional activator. The second type would be usually found in your thyroid hormones, is nucleus bound to DNA and has a retinoid X receptor. Your hormone binds to your corepressor, dissociates. So your receptor hormone complex then activates transcription. So there are also secondary messengers which lead to activation of your transcription factors. Examples are the beta-adrenergic pathway. So this is your increase in cyclic AMP activates your PKA. Then your PKA enters the nucleus and phosphorylates cyclic AMP binding protein. Your CREB is then a transcription activator of genes leading to fuel use rather than fuel storage. So these are the four mechanisms of translation regulation. So the first is phosphorylation of translation initiating factors, translational repressors, disruption of your L4E and L4G interactions, and RNA mediated regulation or gene silencing. So your microRNA silence genes by binding to mRNA, thereby preventing transcription of the mRNA by cleaving it or by blocking it. So some microRNAs are made briefly during development and they are called small temporal RNAs. So any dsRNA that corresponds to an mRNA and is introduced in a cell will be cleaved by dicer into short segments called small interfering RNAs. And this will bind to the mRNA to silence its translation, and this process is called RNA interference. So gene silencing by RNA interference is started again by small temporal RNAs generated by your dicer mediated cleavage with longer precursors. They fold to create duplex regions. The sTRNAs then bind to your mRNA, leading to mRNA degradation or inhibition of translation. Then this dsRNA can be constructed and introduced into a cell. Then the dicer processes the duplex RNA into small interfering RNA, which interact with the target RNA again. Either the mRNA is degraded or inhibited. So ncRNAs also regulate gene expression. This is referred to as INCRNAs when longer than 200. So examples of which would be your HSR1 INCRNA interacting with your EF1A to regulate translation in stress cells. Our knowledge of these ncRNAs is rapidly expanding as further research expands this field. So these regulatory proteins also control development. So remember, embryonic development requires complex and intricate coordination and regulation of gene expression. So more genes are expressed during early development than any other part of the life cycle in a differentiated tissue. So your polarity must be established anterior versus posterior. So for your Drosophila, so metamerism produce characteristic features. So there are homeotic genes. They specify development of organs and appendages, and maternal genes expressed in nurse and follicle cells. So some regulatory process would be the bicoid. So these are transcription factors that activates expression of several segmentation genes, and translational repressor that inactivates certain mRNAs. Or nanos, a translational repressor protein deposited at the posterior of the egg. So this would depend on your anterior-posterior development of your Drosophila egg. So this would be your homeotic genes of your Drosophila. It's often found in clusters and are highly conserved amount in animals. And why is it important? You know that stem cell research is very abundant. So stem cells have the potential to differentiate into various tissues. Totipotent have the ability to differentiate into any tissue. Pluripotent, many types of tissue. Unipotent, only one type of tissue. Such that your embryonic stem cells are pluripotent up till the blastocyst. So adult stem cells would have limited potential compared with your embryonic stem cells. That's why your your placenta is preserved for stem cell. It's your cells are extracted when there is any delivery, but the service is expensive, but it could be done. Adult, limited potential. Its use is for hematopoietic stem cells, gives rise to different cells, and is considered multipotent. Okay? So cells at the morula stage are totipotent and have the capacity to differentiate into a complete organism. So source of pluripotent embryonic stem cells is the inner mast cells of the blastocyst. Then these cells give rise to many tissue types but cannot form complete organisms. So in summary, we have learned the following in this chapter. So positive and negative gene regulation occurring in both prokaryotes and eukaryotes. Examples of gene regulation, specifically the lac operon, tryptophan operon, regulator proteins that contain domains that interact with your DNA, RNA, small molecules, your ncRNAs involved in the regulation of gene expression, eukaryotic gene regulation and development in animals is controlled by cascades of regulatory proteins and their precise position within the embryo. Thank you. I know the lecture is quite long, but there's no other way to go about this. Um, any questions? Um, exam for PMCH1 is deferred. Study for your biochem. We will just have lecture on Friday so that you could concentrate on your biochemistry exam. All right?
Okay po, Doc.
For exam purposes, I'd like you to focus on your lac operon and your tryptophan operon. Of course, other concepts could also be taken from this lecture, but I want you to understand those concepts and the basics of gene expression. Just for me to direct you, I know it's a long chapter, so that you know what to study. Okay? Any questions? Ang hirap, no? Naano kayo? Information overload? If after a while you have questions and need explanation, feel free to chat. I do try to answer. All right. If there are no more questions, you may leave this meeting. You may go back to this lecture or the pre-recorded one, whichever you prefer, in YouTube so that you could study. Okay, I think this isn't included yet in the exam, or included na ba?
Not yet pa po, Doc.
Not yet, diba?
Sa 9th LE pa raw po.
9th lang exam. Alright. So may time pa kayong imbindihin. Okay, thank you for listening to my lecture. See you Friday. biochem exam, and PMCH lecture.
Thank you po, Doc.
Holiday.
Thank you po, Doc.
Thank you, Doc.
Tayong
Doc.
holiday nyo para mag-aral. Sige, goodbye.